Today I'm here to talk about how to leverage spatial technologies to decode tumor microenvironment for biomarker discovery and effective immunotherapies. So until a decade ago, the major tool to study gene expressions or immune responses or signaling pathways was bulk RNA-seq, where the average gene expressions were measured across a large population of cells. But we didn't get information about it's the cell heterogeneity in the cell population. So single cell RNA-seq came into existence, where this technique quantifies transcriptome from the individual cells and really could see how different cells in the same population can act in a different way. Both of these techniques give a lot of information and rich data set to look at the mechanisms of action, but during the process to make the libraries and do the sequencing, we lose spatial context of the tissue. So the spatial transcriptomics methods were developed, where the transcriptome of a tissue could be analyzed from the intact tissues itself. So this became a part of spatial biology revolution. So what is spatial biology? Spatial biology broadly refers to analysis methods that preserve information about the spatial organization of biological specimen or system. So by definition, this technique is not new. We have been doing H&E staining, different kind of immunohistochemistry or immunofluorescence staining for a long time now. So what is new? The new is the level of resolution that these techniques can get down to the single cell resolution and even subcellular resolution, the type of information that we can extract from these technologies, and the amount of data that we can store. So together, these advances have allowed us to generate and analyze large amount of spatial data sets and see how molecular and cellular scale features give rise to important biological outcomes at organism scale. And this information could be very useful to provide insight into how tumors are organized to resist immune surveillance or the mechanism of action of therapeutic agent. So moving forward, how our core can aid into this spatial biology studies. So we have spatial biology platforms that can generate multimodal data. So we can generate spatial transcriptomics or multiplexed imaging data from clinically annotated patient samples or customized mouse or human panels for downstream analysis. So for example, you can have annotated H&E staining slide and see which regions are interesting to you. And then you could do a spatial transcriptomics analysis on it for RNA or protein. You can screen for biomarkers, like an unbiased way of looking at them. And then finally, you can narrow it down to a small set of proteins that you want to validate at a single cell resolution. For that, you can exploit our new PhenoCycler fusion platform that is a very hyperplex immunohistochemistry. So once we have done those assays, the next step is to analyze data sets with a single cell precision. So our core can help you in segmenting and phenotyping each cell in the tissue based on the assessed biomarkers. And you can explore spatial relationships between these various cells by clustering the cells, phenotyping them, doing neighborhood analysis on them, and pathway analysis. What all of this information can give you is you can integrate the multimodal data sets to obtain high quality insights, which you can further evaluate to find mechanism of action, explore relevant biomarkers, and stratify patients or mouse groups based on their cellular phenotypes. So there are a lot of platforms out there. And the first question that comes is, what is best for you? What do you want to choose? So that really depends upon your research goals. Like, do you have antibodies available for your tissues or not? You're doing mouse or human, or the samples that you have are frozen tissue sections or FFPE tissue sections. Are they very precious? They are within the, a window where you should have, like they are better preserved or not, and things like that. And the next thing is spatial resolution requirements. do you really need a single cell resolution for this experiment, subcellular resolution, or is it okay if you look at kind of in a bulk RNA-seq fashion, but in the spatial context? The next thing is, as I told you, the property of tissue or samples. What kind of tissue type is there? Some tissue types are better suited for different platforms. And the last thing is throughput, how fast you want your data. Some of this platform can process like 4 slides at a time, but other can only do 1. And some involve like sequencing timings and all those times. So the turnaround timing really becomes more than the other platforms. So it's really about fine-tuning depending upon whether you want like thinking about cost, time, resolution, and what's best for your research study. So before I move forward to the other slides, I just want to let you know that the way I'll show you here examples is first I'll show you a published paper that came last year, where we will they showed like both the GeoMx DSP spatial platform and PhenoCycler platform that we have in our core. So I thought it would be a good example where you can see how they started with a big data set to screen biomarkers and then narrowed it down to a smaller set of proteins and validated it. So the paper I'm showing for the spatial omics technology integration example is this one, where the authors have looked into the tumor microenvironment of head and neck squamous cell carcinoma in the patients treated with immune checkpoint inhibitors, immunotherapy. So the goal of the paper was to identify protein biomarkers that are informative of immunotherapy outcomes in mucosal head and neck squamous cell carcinoma. So for this study, they used the GeoMx DSP platform and PhenoCycler fusion platform to explore the tumor microenvironment and characterize it at the cellular architecture level. They did both of these experiments on 17 patient samples. The patients were treated with pembrolizumab or nivolumab and categorized based on response to the therapy. And they divided it into 4 categories, depending upon whether they had complete response, partial response, stable disease, or progressive disease. So just to show you how these 2 platforms broadly work. So on the left-hand side, you can see the GeoMx DSP platform where in this paper, so GeoMx DSP can do both RNA assays and protein assays, but in this paper, they have used it to look at the protein expression. So for GeoMx, first step is in the tissue sections, the RNA probes and the morphology markers or antibodies for the certain types of cells that you want to look at, they are added. Then they are fed into the machine and the region of interest, ROIs, are selected. And once the ROIs are selected, the oligos that are bound to the RNA probes are collected in the 9-6-well plates, and they are further processed for library and sequencing. And then finally, you analyze your data. On the opposite side, PhenoCycler fusion, on the tissue sections, first a cocktail of antibodies is added, and then the stained tissue section is loaded onto the machine, where through a microfluidics platform, 3 reporter dyes are added at a time. And this process goes through cycles. And finally, what you have at the last step is like a stitched image with, for all the biomarkers or all the protein markers that you added the antibodies for. And then finally, the analysis is done. So here showing what they found out in the GeoMx DSP platform by using the immuno-oncology proteomics panel of 68 proteins that they performed on the patient responders and non-responders. So here in the panel A, you see the differentially expressed proteins in the tumor samples of both of these tumor section of these patients. And you can see that a lot of immune checkpoint molecules were upregulated, like PD-L1, VIM, SMO, and others. And a lot of molecules like VISTA, FOXP3, CD66P, and PAN-CK, they were downregulated. And you see a similar pattern in stroma. They did the similar kind of analysis on the subgroups, like all the complete response, partial response, stable group, and the progressive disease groups. And what they found was they always see the upregulation of immune checkpoint inhibitor molecules and TNF superfamily molecules. So they hypothesized that if they would make some kind of immunotherapy where you could target those immune checkpoint inhibitor molecules and at the same time have a conjugated protein that can act as an agonist for increased immune activation, it can provide for a better immunotherapy. And how they came to that conclusion, I'll show you in the next slide. So once they had that large data set for protein expression, and they found, they screened for the biomarkers that they were looking for, they narrowed it down to a 45-plex panel on this PhenoCycler fusion, where this platform gives a single cell resolution as opposed to the GeoMx that we previously saw, which was very ROI based, like region of interest based. So you could really not see what's happening in the whole tissue section. So that was their rationale to look into this platform. So for this, they used markers for immune cells, myeloid or structural cells, or the immune activity for T cells. So the PhenoCycler platform could really show a very distinct feature of each treatment type. So you'll see that complete response had like high infiltration of immune cells as opposed to all the other three categories. And if you take broadly, complete response and partial response had way more immune cells than the stable disease or progressive disease. So if you look at the percentage, there was a large amount of B cells and T cells in the complete response group. And this was further shown in a different way, where they showed the tumor heterogeneity in all the four groups in a different way in this figure. So what they really found out that and hypothesized that the presence of these B cells and mature tertiary lymphoid structure in the tumor microenvironment can be an indicative of a better immunotherapy treatment. So just to come to a quick conclusion, what authors found was the compartmentalized expression of immune checkpoint molecules may stratify patient responders from non-responders to immunotherapy. And further sub-analysis of the clinical groupings, they narrowed down that TNFR superfamily of molecules were the important factors in response to the immunotherapy. And finally, this that they could conclude how targeted spatial proteomic approaches may provide new cues to identify biomarkers for immune checkpoint inhibitor therapy in the head and neck cancer. So this was a published study, but now I'll show you examples for the data that were generated on these platforms in the core. So to give you a quick overview of how this DSP platform is run at IAC and what is the cost breakdown. So the assays available are the whole transcriptome atlas for mouse and human, the cancer transcriptome atlas for mouse and human samples, and mouse and human proteome assays. Recently, they have launched a human whole transcriptome atlas and proteogenomics, which can do RNA and protein assay on the same slide. To give you a cost estimate per slide for a GeoMx run is $1980. And before you do an actual run, we like to do a test run, which is about $690. So you get all kind of consultation and panel design support for this. And all of these steps here are staff assisted. You just need to drop off your slides and collect the data. So this was about the GeoMx platform. Next, I'll show you an example for the PhenoCycler platform that was generated by Lepool Lab and Balistnikova Lab. So before, we would have leveraged the current platform that we have, the Vectra-3 staining, which could do like 6 markers. This is an opal dye-based method. But since we wanted to look into a more extensive manner, they utilized, of course, the PhenoCycler fusion platform that can go from 1 marker or looking at 1 protein to 100 proteins. It does it by using the antibodies that are barcoded. And when these antibodies are added to the tissue sections and then finally loaded to the machine, the machine adds reporter antibodies, which is complementary to the barcodes to the antibodies that are binding to the tissue sections. And finally, when they are imaged, you can have a whole overview from the whole tissue section of all different kind of biomarkers that you're trying to see. So just a quick overview of the PCF workflow. First step is validating and optimizing the antibody panel. So here it's written Codex because it was called Codex before. Next step is you add the cocktail of antibodies on the tissue sections, and then you load into machine with a plate of reporter dyes. So what machine does it adds 3 reporter dyes at a time. It will go and image for those 3 markers and then remove the reporter dyes and then add again the 3 different reporter dyes. And this, it goes on in a cycle until all of the data is collected. And finally, you get like a staged image that can be analyzed further with different kind of data analysis platform that I'll let you know in further slides. But I really want to give you more information about this PhenoCycler platform because it's new in the core and not a lot of projects have been done. We have done a couple of projects in the past, and we are looking forward to support more projects. So what is really advantageous for running this PCF platform in the core? So of course, the cost savings comes first. The initial instrumentation purchase, which is a big step. You don't need to worry about the service contracts. We stock kits and reagents. We have dedicated space, tools, and personnel for that. And there is a shared antibody stock that we can provide to the researchers. With that, we have expert resources for staining, imaging, data analysis, assistance with experimental design, troubleshooting whatever issues might come in, and maintain the system well. And then we are trying to develop this antibody conjugation process also, so that if you have some kind of custom antibodies that you want to include in the core panel, you can give it to the core and core can develop it. Otherwise, you have to send it to the company, Akoya Biosciences, and they charge a lot for conjugating those antibodies. So for a mouse, for cost estimates for a mouse frozen section, the cost could be $1,400 for a 12-plex panel. It would take a runtime of 7 hours. And this cost I'm telling you is including the reagents, antibodies, everything. And a human FFPE panel can cost you $1,800 per slide for a 12-plex panel. So all you have to really do is drop off a slide with your tissue sections, and then rest everything can be processed and analyzed by the core. So I know this looks like a lot for processing of this, hyperplex, multiplex immunohistochemistry, but really, if you want to do it in the lab, you have to buy all of these kits, reagents, buffers, antibodies, which are very costly. So if we do it like in a more collaborative way by sharing antibodies, the cost per section would come down to a more reasonable amount. And this is also a good platform because you can fit a lot of a number of tissue section on a single slide. So cost per section goes really down. This is a list showing you what are the antibodies that core have bought to support the research projects. So we have antibodies for human FFPE sections. We have 21 antibodies for that. And then for mouse frozen sections, we have 13 different kinds of antibodies. And this image is just showing you what you can fit in a tissue slide for your PCF run. So it's a big area. I would say you could fit more than 6 sections. They're not too big. Other major services that are provided by the core are the 1 to 4 colors ELISpot or Immunospot. This is an upcoming service. We just bought the instrument and we are going to have a training soon. And then we'll open up this service line for everyone. The other thing is our 24-color spectral flow cytometry, and we provide immunostaining for the cells. Support for this, we have automated immunohistochemistry panel. If you have used our Vectra platform, you already know that. Then we have single-cell RNA-seq platform from 10x Chromium. Then we have MSD multiplex ELISA for multiplex ELISA that can go up to 20-plex. And then tissue dissociation, obviously we have a lot of consultation and suggestion and recommendations. And then we do data analysis for a lot of our services. So just snapshot, what our service turnaround time is. So we have 60 services in total. Turnaround time can range from 15 minutes to 3 days, depending upon what kind of service is being used. We do process a large number of samples at a time. We discuss with users how to customize their projects and what are their specific needs. We do have full service capabilities where we can take a project from start to finish. And as usual, we have like self services and staff assisted services both. This is information on how to get in touch with us to discuss about any of the services or questions about the platforms that you might have. Finally, if you have any questions, I would love to take them. Thank you.
You can say how many tissues you think you can fit for each one of the slides. They're a little bit different, right? The area on the
For
slide.
PhenoCycler? Yeah, for nanostring, since we have been doing it for 2 years, we now know that you can fit like 6 mouse brains there. PhenoCycler fusion, we are still in the early stages, but we know you can still fit, I would say, 6 mouse brains for sure. And if you have skin samples, many, many more. Tumor microarrays have been a big area where people have been using nanostring really. So currently we have been doing a lot of projects on tumor microarrays, and you could put like 70, I would say, 70 cores on one slide. So yeah, a lot of them. For the PhenoCycler also, yes. And PhenoCycler gives really good data because the background is really less, antibody binding is really specific.